Hello. So we meet again. Uh, this is lecture number 7 in the lecture series. So far I told you how you can clone different fragments of a genome in a cloning vector using restriction enzyme and DNA ligase. Right now, this method has a problem. That the problem is that whatever, like say, the particular gene that can rescue the phenotype of a gal mutant strain not being able to grow on galactose as the sole carbon source. Say that particular gene has restriction site. Say you have been using EcoR1 BAM-H1, say it has either an EcoR1 or a BAM-H1 or both sites in the middle of the gene sequence. You do not know a priori, right? You do not know which gene is that you are looking for. And therefore, that gene fragment will never be cloned intact, or there is very little probability of it being cloned intact inside of a recombinant plasmid. Okay. So, and there are many other such challenges. So, to overcome that, a new method has been in practice for quite some time now. I would say at least 16, 17 years, which is called topocloning. Topocloning takes advantage of a bacterial enzyme called topoisomerase, which is a site-specific recombinase. So all that is done is that the vector, the entry vector, will have two sequences, which is the recognition sequence of the topoisomerase and the DNA fragment that you will be generating, this time through polymerase chain reaction or PCR. There in the primers, you will have the same recognition sequence, and then it will go inside the entry clone just like that, without any need for any restriction digestion. And once it is inside the entry clone, then it offers a whole lot of versatility and ease of transferring that DNA fragment to all kinds of different DNA plasmids. I would request that all these different kinds of plasmids that have been shown on this slide, you do Google search to figure out which one is used for what purpose. I don't want to get into all the details of it. Some of these are, very, very important applications. So you can do it yourself. Just use Google search. I'll give you a quick tour of it. Like a 2-hybrid plasmid is required for finding out interacting partner of a particular protein. A mammalian plasmid is if you want to express the gene in a mammalian expression system. Insect plasmid is similarly for the same purpose. If you want to express your gene of interest in an insect cell line, typically the cell line that is used is derived from mosquito and SF9 cells. If you are trying to produce large quantity of eukaryotic protein where appropriate carbohydrate modifications you want to be there, then this is the cell line of choice instead of yeast, where most of the carbohydrate modification enzymes are absent. Or you can express it in yeast, depending on your purpose, or you can express it in E. coli. This whole expression business, I will come to it a little later, okay? Perhaps not in this lecture. Now that, this is how it is done, you can read about the detail of it. These are, this is another variant of it. You use the fudge integration into the bacterial genome. You can use that mechanism. If you read about it, you will see that there's this at P and at B sites. At B site will be on the genome of the bacteria, attachment B bacteria, and at P will be in the plasmid, in the phage, attachment site P. There is also a site-specific recombination. I just don't want to get into it. This cCDB is the toxin gene. If anything is cloned and then the cCDB is replaced by a gene of interest, then the toxin gene is no longer in a plasmid. Therefore, the original plasmid bearing bacteria cannot grow because it will make the toxin, and the recombinant plasmid will grow. Okay, that's pretty much what I would like to tell you here. The rest you study on your own, please. Okay. Now, let's come to the crux of the matter, that you want to test, you want to identify the gene that is mutated in a particular yeast strain, because of which it cannot grow in presence of galactose as the only carbon source. Okay. So, as we discussed in the beginning in lecture 6, that you have to introduce every gene of yeast one at a time in that mutant yeast cell and see which one rescues the phenotype, and that's how you will get it. Now, there is a problem, okay, that you need the corresponding protein to be made, right? It is not enough to have bits and pieces of the genomic DNA. You need to have all the elements of the genomic DNA so that the protein is made. Now, this is a problem for most eukaryotic genes because there is very large introns inside of a gene, and these introns are not part of the sequence that codes for the protein. So for those kind of for this purpose, therefore, instead of the genomic DNA fragment, what is done is DNA replica of the messenger RNA or mRNA is cloned inside of the plasmid. Now, this will require that you isolate all the mRNAs of yeast, okay, or any other organism that you might be looking at or you have been working with, and then you have to make the double-stranded cDNA replica for each one of these mRNAs, and that you have to clone inside of a vector. And please remember that your end objective is to produce the protein for the particular gene. Okay, therefore, just having the DNA sequence for the mRNA is not good enough. You have to ensure that that DNA sequence is transcribed and translated. Therefore, the plasmid must have DNA elements that will allow transcription and translation of the insert in the recombinant plasmid. For that, different elements are needed. I will come to that later when I will explain to you the features of an expression host. Okay, I will come to that a little later. But thankfully, when you are dealing with yeast, you do not have to do this. Yeast, for most of the yeast genes, yeast Saccharomyces cerevisiae, not Pombe. For most of the yeast cerevisiae genes, there is no intron. Therefore, the coding sequence is contiguous, which is not the case in flies or humans or even in Saccharomyces pombe, Schizosaccharomyces pombe, but the coding sequence is interrupted by introns. Okay? So for yeast, genomic DNA is as good as cDNA library when you are talking about rescuing a phenotype. So you can potentially clone a whole lot of, like every piece of yeast genome in different plasmids, and you will have to ensure that the total plasmid in your collection and the insert that they have, the total length of the insert in all these plasmid collection adds up to 2 to 3 times the genomic DNA length. Okay, like I do not remember the length of the yeast genome. I will inform you in the next lecture. I do not want to interrupt this. Okay, so let me just pause the recording for a minute. Let me figure out the genomic. Okay, so yeast genome is 12 megabases. Okay, yeast Saccharomyces cerevisiae genome is 12 megabases. So if it is 12 megabases and average length of a fragment that you have generated by double digestion is say 1200 bases. Okay, I'm just going on the lower side. It is in general you go for 3000 bases. Let's say 1200 bases. So therefore, in 12 megabases, which is 12,000 times 1000. So if you have 1000 times 10,000 individual recombinant plasmids, you should be able to cover the entire genome at least once. Okay. Now the problem is that how is the experiment done? You take a whole bunch of yeast cells, typically a pellet that will have few billion cells. Then you lyse it open, then you digest the DNA, and then you do the cloning. So in reality, the genome of yeast is not represented once, but few million times. Okay. Therefore, the number of recombinant plasmids that will be produced will be 12,000 times few million. Okay. So if you take randomly any 12,000, the probability that you have covered the entire genome at least once is very, very slim, extremely, extremely low. So typically you go for much higher number, okay? Like I would say that, you go for a few million at least, okay? So if you go for few million recombinant plasmids, chances that you have covered the entire genome at least once is very high. Okay, so it is important that you cover the entire genome at least once, because you are looking, you cannot afford to overlook any one part of the genome. What if your gene of interest is right there, like the gene that can rescue the phenotype is right there. So therefore you have to go this little extra mile. So, and as I said, that here when you are trying to rescue the phenotype, you have to ensure that the protein for the corresponding gene is expressed. Therefore, you have to have the machinery for the protein expression to be there. So for that purpose, what you do is you use something called a shuttle vector. What is a shuttle vector? A shuttle vector must have the ability to replicate both in two different organisms. The two organisms between which the plasmid will be able to shuttle. And in almost all the cases, one of the organisms is bacteria E. coli, and the other one, depending on what it is. So here, you see in this plasmid, you have this ORI, which is the ORIC, or origin of replication for bacteria. And then you have this ARS, autonomously replicating sequence. This is the DNA element that ensures replication of plasmid in yeast. Okay. Then in yeast, unlike in bacteria, you want the plasmid to be equally distributed in both the daughter cells, as the cells will be multiplying after transfer infection, like when you first introduce the plasmid inside the cell, the next time cells divide, what if one cell does not get the plasmid? Then, like that, in that cell, the rescue just cannot happen. So you have to ensure that the plasmid is inherited in both the daughter cells. For that, you have another sequence called the CEN sequence, or centromeric sequence, which ensures that at the end of mitosis, the plasmid is distributed to both the daughter cells. Okay, and then the ampicillin resistance is for selection in bacteria, and URA3 is for selection in yeast. In yeast, antibiotic selection is used less frequently. More frequently, what are used are different kinds of genes that make one or the other critical nutrients. Okay, so URA3 is one of the genes that is essential for making uracil. So the host yeast cell will be mutated for this particular gene. So it will be URA3 minus, like in yeast, we write like this. So the host yeast cell will be mutated so that it cannot produce functional URA3 gene, and that functionality will come from the plasmid. And therefore, only in the yeast cells bearing the plasmid, they will be able to grow in a medium that does not have uracil supplied in the medium. Okay, so this is what is referred to as a shuttle vector, and then of course you have the polylinker sequence, that is your multiple cloning site, where a piece of the genomic DNA is integrated. So this is what is shown here, that you have the yeast genome, you digested something like SAL3A, which is a like a tetracutter. You'll typically get around 300 nucleotide or so fragments. I would prefer to go for a little like at least a hexacutter, and then you clone those in similarly digested shuttle vector, and then you are going to generate all different kinds of recombinant plasmids. Recombinant because they will have the shuttle vector, and in the polylinker site there will be a sequence of DNA which is a piece from the genome of yeast. It can be any one of the the 10,000 pieces from yeast genome, and then you will transform the mutant yeast cell. This yeast cell cannot grow in presence of galactose as the only carbon source, or if you remember the temperature sensitive mutation for cell cycle, the CDC genes, cell division cycle genes. So at a higher temperature, it cannot grow, and then you transform that temperature sensitive CDC mutant or gal minus mutant with the yeast genomic library. Again, the same principle operates, that in every yeast cell, one and only one kind of plasmid can go in. That is depicted here, that the yeast cell that is transformed is either blue or red or green or orange, like these four colors. Okay. Now this plating is done in permissive temperature. Okay, because at permissive temperature, these cells will grow no matter whether the defective gene is supplemented from the library or not. Okay, so this is called a library, a collection of all the recombinant plasmids, each having a different fragment of yeast genome. That whole collection is called a library. But when you should not get the image of a shelf of plasmids. A library is typically in a tiny plastic tube, which we call Eppendorf tube, and approximately 200 microliter in volume, and that is concentrated DNA solution, where you have a small circular plasmid DNA, recombinant plasmid DNA. Each of the plasmid DNA will have a fragment of the genome, and it will the same genomic fragment containing plasmid may be in multiple copies, but efforts are made to make sure that in the overall collection in the library, every fragment of yeast genome is represented at least once. Okay, you do the transformation. So in a typical plate, you can grow maybe 250, 300 plasmids. Well, 250, 300 yeast colonies, so that the colonies can be visualized distinctly. So 4 such plates will be 1000. Your idea is to go for 10 to the power 5 clones. Okay, so you will go for 400 plates. Okay, so you transform the yeast cells. Say the transformation volume is 1 ml. Okay, so 1 ml divided by 400 is 2.5 microliter. So 2.5 microliter you will be plating on one plate. Okay, so that way you will be plating 400 different plates. Each of the plates you will grow them in 23 degrees centigrade, replica plate it, then put one replica in 23 degrees centigrade and the other in 36 degrees centigrade. And then you will see that at 36 degree, most of the colonies do not grow. Remember, all these colonies are coming from only one strain. That one strain that you have identified as a gal minus or CDC28 or something like that. So none of these are supposed to be. So this is strain cannot grow in at 36 degrees centigrade, or if it is the gal selection, then it cannot grow in presence of galactose as the only carbon source. But if it has received the right DNA, in this case it is shown as this red colored genomic fragment bearing plasmid, if it has received that one, then and only then it will grow. Okay, so you know that, look, none of the other colonies grew, but these two colonies grew. So now you know that these two colonies are harboring the plasmid that is complementing the yeast gene that is mutated. So there are methods of isolating the plasmid from the yeast colony and by simple DNA purification. And then once you have purified the DNA, you sequence it, and you sequence it and you get the sequence of the gene that is mutated in the original yeast strain, because of which it was a temperature sensitive mutant. Okay, so now you understood the whole thing, right? Let me quickly recap from the very beginning that how do you identify a gene involved in galactose metabolism. So you take a wild type yeast cell, haploid, you treat it with one of the many mutagenizing agent, so that DNA sequence changes. Only one sequence change occurs in one yeast cell at a given point of time, and then you have some millions of mutant yeast cells. You plate them on yeast medium, and then you grow them at 23 degrees centigrade. After they have grown in 23 degrees centigrade, you make 2 replicas. Okay? Replica meaning Xerox, photocopy. Okay? You make 2 replicas. It is not really photocopy in that sense, but the colony positions will be identical to the parent plate. And then you put one plate in 23 degrees centigrade and another in 36 degrees centigrade. Then you will see which are the colonies that grew at 23 degrees centigrade but not at 36 degrees centigrade. You let us stick to the gal minus example, galactose metabolism example. So you plate one replica on a plate containing glucose and the other on plate containing galactose. The ones that did not grow in presence of galactose as the sole carbon source are the mutants in galactose metabolism. You do not know where the mutation is. Then you do complementation analysis to figure out how many complementation groups are there. The number of complementation groups give you some idea about the number of genes that might be involved in this particular pathway. Then if you want, you can do epistasis analysis to arrange the genes in some order of action in the galactose metabolism pathway, and then you take any one of these mutants and you want to figure out what is the actual gene that is mutated in this. So you construct a genomic DNA library, transform the mutant with that library, and you figure out the colonies that are rescued because of this genomic DNA transformation. You get that particular strain where it is rescued, isolate the plasmid from there, sequence it, and you get the gene. So if you have 5 different mutations, you can do that for 5 different genes. Now you want to know that if there are other genes in the pathway that you may have missed, and it is possible that they may be functionally interacting, functionally or physically interacting with the gene that is mutated in this particular strain. So you can do a synthetic lethal screen or a suppressor screen, either of which will give you, other genes that are interacting with your gene of interest, the original gal minus mutant, either physically, like forming dimer, or genetically. If there are 2 different pathways, both are responsible for the 2 parallel pathways, then that is not physical interaction, but there is genetic interaction. So you will be able to figure that out. So this is how you get to know the whole pathway. Okay? When I will be teaching you about cell biology, you will see that how such screens were so effectively used to understand and learn everything about secretory pathways in our cells. Okay, so this is how it is done. Now, as I said, that in yeast you can make do with a genomic DNA library, simply because yeast cell genome, Saccharomyces cerevisiae genome, does not have introns. But what if you are studying Drosophila or humans, or even Schizosaccharomyces pombe, the fission yeast? There the genes have introns. So there, this genomic DNA library will not serve the purpose of rescuing a phenotype, because the actual length of DNA is much longer than, the actual length of DNA is much longer than the length of DNA that is responsible for production of a protein. So chances of having all that DNA, meaning from the first exon to the last exon with all the introns in between, to be cloned in a plasmid, that probability is very, very low. Okay, because often it is in tens of thousands of bases. So in those cases, you need to go for what is referred to as cDNA library. Okay, so as I mentioned earlier, the cDNA library is basically a complementary double-stranded DNA copy of the mRNA. So just like you didn't know which DNA fragment from the genome will rescue the phenotype, here also you do not know which particular mRNA is responsible for the for your gene of interest. Okay, so what you will do is you will take all the mRNAs of the cell and you will make double-stranded DNA copy for each and every mRNA of the cell. Now, how do you do it? All mRNAs, they have one property in common, which is not there in any of the other RNAs, like tRNA or ribosomal RNA. That is, they have a poly-A tail. Because they have a poly-A tail, so if you have a DNA that has only T's, oligo-DT, and it is immobilized on a resin, and you run the RNA solution through it, only mRNAs will be arrested onto that column. Okay, so that way you can arrest the mRNAs, and then you can use poly-T as the primer to hybridize with the mRNA, and then you do a you use an enzyme called reverse transcriptase that can use RNA as a template to make DNA. Okay, it is called reverse transcriptase because normal transcription is copying the DNA into an RNA, but here you are copying an RNA into a DNA. Like there are many ways of doing that. Nowadays, the methods have changed dramatically. But I'm giving you just one example that is there in your textbook, is that you use a oligo-DT primer and you do reverse transcription, and then you make this RNA-DNA hybrid. It's an RNA-DNA hybrid. The red part is RNA, the green part is DNA. RNA is alkali labile while DNA is not. So if you treat it with a very dilute alkaline solution, then the RNA part will be disintegrated or chewed off. So you are left with only the DNA. To that, you add a set of G's in a string. Okay, it's another artificial DNA, just poly G, and that will be ligated here. Okay, once that is ligated here, then you can use a poly C as another primer, and you can do a PCR this time, and you will get a double-stranded DNA where this extra sequence of G's and C's are there on one end, which you actually artificially created by doing this part. Okay, so now this is your double-stranded DNA, and because these are primers that you have designed yourself, so you can then add any piece of DNA sequence to it. Often we use what we call a linker, which is basically nothing but a small double-stranded fragment of DNA bearing any restriction enzyme of your choice. So in this case, we have appended EcoR1 sequence to it, and then you digest it with EcoR1, and you digest expression plasmid, yeast expression plasmid, or mammalian expression plasmid, and you clone it. And again, you have to be careful. Okay, so now let me give you two different things. That is, making a cDNA library is much more tricky than making a genomic DNA library. I will repeat another time. It is tricky because in the genomic DNA library, you already have the polymer. You are only cutting it in small pieces and packaging it in different plasmids. But in this case, you have to first make a DNA from the RNA. Now, RNA, because it is single-stranded, unless you absolutely need to, like you have not studied it in your undergraduate degrees or you have no idea, I will prefer not to get into that discussion. But this should be sufficient if I tell you that unlike DNA, RNA has a complicated secondary structure because it is single-stranded. Okay, so it satisfies its hydrogen bonding needs, unlike in the DNA, which is done through formation of the double-stranded molecule. Single-stranded RNA, it satisfies its hydrogen bonding need through intramolecular hydrogen bonding, and that gives you this secondary structure. And some of the secondary structures are very stable and stubborn. So when there is reverse transcriptase, so imagine that this is the RNA, this is your the 3' and this is your 5', and the reverse transcriptase is going through it, and suddenly there is this secondary structure. So reverse transcriptase will get stalled here. It cannot bypass this part. Okay, it cannot bypass this part. And therefore, your cDNA formation will not be complete. Okay, sometimes because the RNA is very labile, so sometimes full-length cDNA becomes a problem. So there are many challenges, and there are many experimental ways of minimizing these challenges. I would not say eliminate, but minimizing these challenges. That is one problem. Okay, that depending on the length of the cDNA that you are looking for, it can be a challenge if it has very high GC content. Okay, like just to give you an example, I work with a gene that whose mRNA has 88% GC content. It is so difficult to work with that you cannot even do a reverse transcription. Forget about cloning or manipulating. Nothing happens. Okay? So that is one challenge. The other challenge is representation in a population. I will go a little slow. Think about it. Okay, how is it done? How is the experiment of reverse transcription PCR cloning done? You have few million mammalian cells. You add, hypotonic buffer. The cells will burst open, and you will isolate the RNA using, one of the chemical extraction methods. So in this RNA solution, you have a true molar representation of the RNA concentrations inside the cell, right? Because you have not done anything to preferentially enrich one type of mRNA versus the other type, okay? You know that certain kinds of proteins are expressed at a very high level, like proteins that are involved in cytoskeleton, actin, tubulin, proteins that are involved in ribosomal, in ribosome, okay? So ribosomal subunit proteins, okay? Proteins that are involved in basic processes of carbon metabolism, like GAPDH, glyceraldehyde 3-phosphate dehydrogenase, okay? This is one of the most abundant genes in our cells. So these mRNAs for these proteins will be present at a very high number as opposed to an mRNA required for a very specific signal transduction pathway or a transcription regulator. Okay, so therefore, all different types of mRNAs, like I will talk about it more when we talk about transcription regulation. But in a mammalian cell, typically it has the ability to make 23,000 different mRNAs. But a particular cell type typically will express some 5,000 of it. Okay, all the cells do not make all kinds of mRNAs. We'll come to that a little later. So, you think of a cell type that makes 5,000 different mRNAs. It is not that if there are 20,000 mRNAs in the cell, each of these 5,000 will be present 4 pieces each. There will be some which are hundreds of them, and there will be some for which there is only one. Okay, so therefore, when you are making cDNA corresponding to the mRNA population, the cDNA population will also be skewed towards this highly expressed mRNAs. Okay, so your cDNA library, unlike the genomic DNA library, will not be equally representative. Okay, it will be biased towards mRNAs that are highly expressed, because that is a problem that you need to keep in mind. So, there are other ways of making cDNA library. This is another different method. I will not get into it. Please look into it, go to the company website. I have provided the trademark and learn more about it. If you have any question, come back to me, ask me, I will explain. Okay. So, no matter how you do your serial library preparation, you will have this problem of uneven representation. Okay, so therefore, if you want to have every mRNA of your cell to be presented at least once, you need to go for a much higher number. Yeah, as I said, typically 5,000 different mRNAs will be produced in a given cell type. But the advice from the experts is that you should go for 1,000 to 10,000 fold excess, like as the recommendation is that if you have a pool of 10 to the power 6 to 10 to the power 7 different bacteria, chances are that one of the bacteria will have the mRNA of your interest, okay? Like, or every mRNA will be represented as a cDNA in the pool if you go for this kind of number, 10 to the power 6 to 10 to the power 7. So what are you doing? You are doing the same thing as you did for the genomic DNA library. You have your RNA, your total RNA pool, you use oligo-DT column to specifically purify the mRNA which have poly A tail, and then you go for reverse transcription to make the first strand cDNA, meaning exact complementary DNA of the mRNA, and then you destroy the RNA, and then you use one of the many tricks to make the second strand, and then you clone it in a plasmid. Depending on what your end application is, it can be a cloning plasmid or an expression plasmid. Okay, so you clone it, and then you transform bacteria, and then you pick individually, 10 to the power 6 to 10 to the power 7 bacteria, make solutions of them, freeze them. This collection is your bacterial cDNA library for mammalian cells. Okay. If you don't want to go there, you can also do it like this, that you transform and you keep the transformation mix as a library. Now let us come to one particular example today, and then I will end this lecture. Say you want to identify you want to know the whole sequence for, like, the mRNA sequence for a particular gene that you are interested in. Okay. What do you know about it? Somehow you know sequence of little part of it. Please forget about it that for most of the genes today you have the whole genome sequenced. Like, I can guarantee that for most purposes you will not have to use any of these techniques in today's lab, but you need to know how they were originally done, so that when you are thinking of a completely new project, you are not blindsided to the historic development of molecular biology that you are practicing today. And if it gives you any solace that I am old, of course, but not very old, I have used these technologies. The first gene that I cloned and I published in 1997, I used exactly this method. Okay, so either through biochemical means you know the protein, and therefore you know the protein sequence, and you have predicted the mRNA sequence. As I said, that if you know a small peptide sequence, you can, by taking codon degeneracy into account, you can predict all sorts of sequences that can be for that mRNA. Okay, using a very simple computer algorithm. Or, I don't want to get into that detail right now. You know a little part of the sequence towards the 3' end of the mRNA, okay, which is referred to as EST sequence, expressed sequence tag, okay, EST sequence. So when you know that, you will make a radioactive replica of that mRNA, okay? And it can be a double-stranded DNA. The radioactive replica can be double-stranded. It typically does not have to be longer than 100 to 100 nucleotides, no matter how long your mRNA is. Okay, 100 to 100 nucleotides. Now you have this 10 to the power 6 to 10 to the power 7 clones. the different bacteria. So you have plated them, and again you make replica plate. Okay, the original will serve as your reservoir where you can go back when you have identified your important clone, and then to again, like if there are, say, 300 colonies per plate, then 3 colonies will have 1,000. Okay, so you are talking about 3,000 to 30,000 plates where you have plated your entire library. Okay. Now you will go in with a filter paper, which will be, when you spread it nicely on the bacterial plate, there will be some bacteria that will be transferred onto the filter paper. And then you put it inside an alkaline solution. The bacterial cell will be lysed, DNA will be denatured, and you dry it. Okay. Onto that, you add this radioactive DNA, which is a little part of the total mRNA sequence. Then, following the methods that were established by Southern hybridization, those parts of the filter where the, like if this is the whole filter paper, which is a replica of the bacterial plate, there were all these different colonies. Only on this colony you had the DNA that is complementary to this. Okay, so this radioactive DNA will hybridize here and not to anything else. So when you expose it to an X-ray film, you will get just one spot on the X-ray film. So if you align the X-ray film to the bacterial plate, you will know which colony it is that has the complementary sequence to this. You get that particular bacteria, you isolate the plasmid from there, sequence it, and you get to know the entire mRNA sequence for your protein of interest, okay, or your gene of interest. So this is how library screening is done. Okay, so I will stop right here. I want you to revise everything that I have taught till this point, because I will now get into a little bit more advanced and recent technologies of transcriptomic analysis, comparative transcriptomics, etc. For that, until and unless you are very comfortable with all these lectures, it will become very difficult. Okay. Therefore, please, study very hard. Not only you listen to my lecture over and over again, also consult various internet sites to learn more about these techniques. As I said, that the purpose is not to prepare you for an exam, not to get you closer to a degree. The purpose is for you to learn how to conduct these experiments yourself. Therefore, unless you are very comfortable with the concepts, you will not be able to come up with an experimental design on your own. Okay, so please invest your time. I can guarantee that you will slowly start to enjoy this whole subject. Okay, so with that, I will end this. Please revise from lecture 1 to lecture 7 thoroughly before we move on to the next segment. Thank you.